Hello everyone, welcome back to this amazing platform of Infinity Learn by Sri Chaitanya. My name is Kiran Choudhary and I am your biology faculty at Infinity Learn. Students, in this session we will discuss very important PYQs on chapter number 6 of your class 12th NCERT biology textbook. Chapter name is Molecular Basis of Inheritance, and let me also clear that we are going to take all important questions that NTA has asked from 2013 to 2021. And also please note, I am going to take this PYQ session on this chapter in 2 sessions. So today is session 1, or I can say part 1, and tomorrow we will do part 2. And also please note that in this session we won't take PYQs abruptly, right? Rather, this session is going to be systematic, where I am going to tell you, as well as I am going to discuss with you the previously asked questions by NTA in Neet topic wise, and the sequence of the topic will be exactly same that is in your NCERT textbook. So same, we will start with the introduction or the DNA structure and packaging part, those topics, and then we will continue forward with the search of the genetic material and replication. So in this class we will discuss all important previous year questions NTA has asked from 2013 to 2021 till replication part, and from transcription till the end DNA fingerprinting will be covered in the next session. So let us start our class, students, and also I would love to tell you, do visit to our website infinitylearn.com and check out the amazing courses that we offer there, and you can use any of the subscription or courses at straight 40% off by applying the code Kiran40. By applying the code Kiran40. So do consider visiting to our website infinitylearn.com and use the subscription or courses at straight 40% off by using the code Kiran40, right? Hope this much is clear to all of you. Hello Safura, welcome to this amazing session. Okay, chaliye start karte hain with the very first question. Yeah, as I told you, this session is going to be systematic, so let us see which questions NTA has asked from the topic the DNA. Will include first of all the questions from structure and then from packaging. So let us see what questions NTA has asked from the DNA structure of polynucleotide chain. This is the question that NTA asked in Neet 2020, very latest, right? So tell me students, I would love and appreciate if you are going to participate in this session, right? So Safura, how about letting me know the right answer to this question? First, let me read out the question for you. Which of the following statement is correct? Which of the following statement is correct? So it's asking about the pairing, right? So see students, DNA is a double-stranded molecule. 2 strands are present in the DNA, where one strand is connected or bonded to another strand with the help of hydrogen bonds, and these hydrogen bonds are formed between the nitrogen bases that are present on these 2 strands, right? So accordingly, you know, there are 4 possible types of nitrogen bases that can participate or that can be present in the nucleotides of the DNA. It can be adenine, thymine, guanine and cytosine, right? So please help me in knowing which one do you think is the right option. So here you need to tell students, adenine pairs with thymine with how many hydrogen bonds. Not to forget students, in a DNA, adenine pairs with thymine and guanine pairs with cytosine. The question is with how many hydrogen bonds adenine pairs with thymine. Can anyone tell me? Super simple question. If you are still unable to get the answer, let me help you with this diagram. Yes, so this is a figure that NCERT has provided you in the structure of polynucleotide chain covered under the topic the DNA, right? And if you notice this, this is what a segment or a part of a DNA, right? 2 strands, right? And what is this? This is a nucleotide. What is this? This is a nucleotide. This is also a nucleotide, and both these nucleotides that are present on 2 separate strands. Hello Sunil. Both these strands, the nucleotides that are present on these 2 strands will be bonded with the help of hydrogen bonds. So see here, adenine is paired with thymine with the help of 2 hydrogen bonds, right? And see here students, guanine is paired with cytosine with the help of 3 hydrogen bonds. So I hope students, with this diagram you will not get confused ever in future that adenine pairs with thymine, A pairs with T with the help of 2 hydrogen bonds, and G pairs with C with the help of 3 hydrogen bonds, right? So let us see now the right answer. Yes, so adenine pairs with thymine through 2 hydrogen bonds. So A pairs with T with the help of 2 hydrogen, and G pairs with C with the help of 3 hydrogen bonds. Perfect. Correct, Safura. Now let us see the next question. Next question is purines found both in DNA and RNA are. What do you think? What should be the right answer to this question, students? You try to think, what is a purine? What is a pyrimidine? And if you ask me about a hint, let me give you a hint, students. Purines, if I talk about purines and pyrimidines. Let us talk in general first. Right, let me write it again instead. So see students, purines and pyrimidines both are present in DNA as well as in RNA. Now the question is asking about, which purine and which pyrimidine, right? So see here, this purine, if I say it is A and G, adenine and guanine are considered as purine. Talking about pyrimidine, there are 3 types of pyrimidine, C, U and T, right? So see students, out of these purines A and G will be present in both DNA as well as in RNA. While talking about pyrimidines, C, right? This C will be present again in both DNA and RNA. But U will be present only in RNA, and in place of U in DNA it will be T. So see students, both A and G, adenine and guanine, are present in DNA as well as RNA. But when it comes to pyrimidine, C is present in both U in RNA and T in DNA, right students? So the purines found in both DNA and RNA are adenine and guanine, right? So now with this question, let me show you also how adenine and purine and pyrimidines look like. So let us take a small revision out here. What is these? These are the 2 purines, and remember students, the purines are double cyclic compounds. Adenine is made up of 2 cycles, as well as guanine is also made up of 2 cycles, right? And talking about pyrimidine, this is how pyrimidines look like, and they all are heterocyclic. Even these were also heterocyclic. They all are heterocyclic and made up of single ring, right? I hope this is clear to all of you, where thymine will be present only in DNA and uracil only in RNA. Next question. This question was asked in Neet 2017. The association of histone. The association of histone H1 with a nucleosome indicates that. What do you think? What should be the right answer to this question, students? Come on, give it a try at least. Safura, Sunil, I want you to participate. See students, let me give you a small hint over here. If I talk about histones, what are histones? Histones are the basic proteins, the proteins that helps in packaging of the DNA in eukaryotic cell, right? There are 5 types of eukaryotic histones. There are 5 types of eukaryotic histones. first H1, no sorry, yeah H1, H2A, H2B, H3 and H4. Now see students, H2A, H2B, H3 and H4 all will occur in pair, all will occur in pair. So there are 4 types, here there are 4 types. When they will be in pair, they will present in total number will be 8. So H2A, H2B, H3 and H4 will occur or will form a complex. That complex will be known as octamer. It will be made up of 8 molecules, 2 of each, right? And such see here, such a core, this is the core, right? This core is made up of the histone molecules H2A, H2B, H3, H4 in pair is going to make the core of the nucleosome, right? Now on this core of the nucleosome, DNA is going to wrap, right? And what this H1 is going to do? H1 is going to seal the incoming DNA and outgoing DNA, right? So this is a DNA that is coming and going to overlap on this core of the core molecule of the nucleosome, which is made up of those 4 types of histone molecules occurring in pair. And what is H1 doing? Sealing, right? Sealing the incoming and exiting the DNA. So this is what this whole complex is known as nucleosome. This whole complex is known as nucleosome, where you can find the core of the histone molecules H1 as well as the part of the DNA that is being wrapped around this core particle, right? So see students, coming back to this question is association of histone H1 with a nucleosome indicates, what it will indicate? Packaging, right? What it will indicate? That the DNA is condensed into a chromatin fiber. So presence of H1 or association of histone with a nucleosome will actually indicate that yes, now the double helix of the DNA is now going to be packed, ultimately into a chromatin and even beyond that. So I can show you students, see how the packaging work, right? So right now with the help of this image, I will try to explain you the levels of packaging, right? So with the help of this effective diagram, let us try to understand what are the various levels in which the double helical DNA is going to be packed. So first of all, it is double helical, right? Next stages, it will be packed in form of nucleosomes. From nucleosome, it will be packed. See this DNA helical is getting packed around, right? Packed around these histone molecules, right? So there will be formation of nucleosomes that can be seen as a bead on string structure under an electron microscope. Further, these nucleosome, my kids, these nucleosomes are going to be packed in a solenoid. What is this? They are going to be packed in a solenoid. Again, this solenoid will be packed in another complex form which is known as supersolenoid. From a supersolenoid, there will be formation of chromatin. Right? Then again, the chromatin will compactly condense to form chromatids, and from chromatids finally what you will get, instead I should write chromosome here, not centromere, okay. So I wanted to make out a point, I wanted to point out the chromosome here, right, chromosome, right. So it started with double helix DNA, then to nucleosome, then to solenoid, supersolenoid, chromatin, chromatids, and finally into a chromosome, that is the most condensed form of or most packed form of DNA. Is this clear? Hello Shubhika. Okay. Thank you Shubhika. Thank you for the appreciation. Next. I hope this question is clear. Now let us discuss, this question was asked in 2015, although this exam was cancelled, but question is a question, right. Next, in a sea urchin, the DNA which is double stranded, right, double stranded, there is 17% of the bases were side to side. The percentage of the other. Now remember students, when it is a DNA and also a double stranded one, there will be A, T, G and C, right. In this question it is stated that side to side C is actually 17%, and this question is asking you need to tell the percentage of A, T and G present in this entire length of the DNA of this particular organism, right. So let us see this, how to calculate this. So my lovely students, let me give you a tip over here, how to solve these kinds of questions. Tip is very simple. What you want to do is you need to remember Chargaffs Rule, or you need to remember complementary base rule. Very simple. You need to remember complementary base rule. What complementary base rule says? It says A will pair with T and G will pair with C. I repeat, for a double stranded DNA molecule, A will always pairs with T with the help of 2 hydrogen bonds, and G will always pair with C with 3 hydrogen bonds for a double stranded DNA molecule, right. Same thing you need to follow. So my students, let me also give you this recap. Remember, from this only I concluded that A pairs with T, G pairs with C, right. So that means in that entire length of the DNA, wherever there is C, in front of that there will be G, right, and wherever there is T, there will be A, right. So the question is 17% is side to side. Simple. That means, and you already know that G pairs with C always in DNA. So wherever there is C, there will be G in front of that. So similarly, if there is 17% C, that means there should be 17% G also. They cannot be extra or less G, because if it is so, it will pair with what? The structure of the DNA will get disturbed, and that should not happen, and that never happens, right. So the percentage of G and C will remain same, hence therefore C, because G pairs with C, therefore percentage of G1I will be 17%, right. So if you sum up, remember students, Chargaffs Rule. What is that? Chargaffs Rule, very, very simple, right. So see students, if this is 17%, so already we know G is 17%. Now we know C is also 17%. The total number of bases, the total number of nitrogen bases in a DNA can be 100%, because we are taking percentage over here, maximum can be 100, right. So if I consider the total, it will come to 34%. So if I consider G plus C, so total sum of G plus C will be 34%. Now out of 100, how many is left? Percentage of nitrogen bases left is 34, 100 minus 34, that is 66%. That means in 66%, who is present? It is T plus A. It is T plus A. So remaining 66% will be formed by these two togetherly, these two nitrogen bases A and T. And you already know that A always pairs with T. That means the number of A, percentage of A will be equals to percentage of T. Hence, you can consider them equal, or you can divide 66% by 2. And finally, what you will get? 33%. So 33% will be T as well as 33% will be A. So 33A, 17G and 17C is the correct answer. Very right, Safura. Why are you deleting the message? It is correct. It is accurate. Don't worry. Next, let me give you the answer. It should be 17G, A33 and T33. Alright. Next question. It was asked in Neet 2015. Which of the following is applicable to RNA? Can anybody tell me which of the following is not applicable? Which of the following is not applicable to RNA? Can anyone tell me? Come on, give it a try at least. Give it a try at least. Give it a try at least, right? So, let me tell you, students, Chargaffs rule. Chargaffs rule are applicable only on double stranded molecules. Are applicable only on double stranded molecules, and this is a universal fact that RNA is single stranded. How Chargaffs rule will be followed on them? Let us see the other option, heterocyclic nitrogen bases. It is present in both DNA as well as an RNA. Complementary base pairing, yes, complementary base pairing happens in RNA, right, when DNA needs to be, or information from DNA needs to be copied in form of RNA. Yes, so RNA also follows this complementary rule, right. So, this is not something which is very strange over here. So, complementary base pairing is also followed, and you remember just in G pairs with C and A pairs with U instead of T in RNA. Next, 5-phosphoryl and 3-hydroxyl ends, they will also be present because these ends are with respect to the single strand, right, they are not with respect to two strand. So, each of the strand of a polynucleotide chain will contain 5-phosphoryl and 3-hydroxyl group. So, all these are possible, but Chargaffs rule is for only double stranded molecule. Very right, Safura. Let us see the next question. Yeah, let us take a brief revision of what Chargaffs rule is. So, see students, what Chargaff, let us take a quick revision, okay. So, in Chargaff says purines will be equals to pyrimidines, right. In a DNA, in a double stranded DNA or in a double stranded molecule, purine will be equals to pyrimidines. The amount of purine will be equals to pyrimidine. Hence, what is purine? Purine is A and G, and what is pyrimidine? T plus C. So, A plus G will be equals to C plus T according to this rule. So, there is deduction from this one only. Second, now if you send this C plus T in the base, what will happen? A plus G and denominator, in denominator you have to put this. So, ultimately you will get this third point, A plus G divided by C plus T equals to 1. Yes, because this is reduced from this and this one is reduced from this one, right. So, this is the result of this one, and obviously the answer should be, the fraction, the value should be 1, and this value is constant for all the species irrespective of any. So, there are no exceptions to it, it is constant for all species. Second is the base ratio. So, the ratio of A plus T and G plus C, it is constant for all species. It will be constant for human, constant for buffaloes, constant for E. coli, constant for, right. So, whosoever is possessing a double stranded DNA molecule, it will be constant for a species. The ratio of A plus T and G plus C, this ratio is a bit different from this ratio. Please notice, this is purine upon pyrimidine, but here purine and pyrimidine both are present. Please notice this, right. Simple. Thank you Sunil, thank you Safura, thank you Shubhika. Let us see next question. The diagram shows an important concept, very important. This question was asked in Neet 2021 latest, Neet 2021-23, and let me tell you one more thing students, NTA till now has asked this question, this particular format of this question or this information in this format, in this particular format at least 3 times. Yes, at least 3 times NTA has asked this question, right. So, it becomes important and it is super easy, super easy. You just have to follow what? Central dogma. It is based on central dogma of molecular biology, where it says when DNA, information from DNA is copied to DNA, it is known as when information from DNA is copied to DNA, it is known as replication. It is known as replication. When genetic information is carried from DNA to mRNA, it is transcription. It is transcription. When genetic information is carried from RNA to protein, it is translation. It is translation. It was proposed by Francis Crick, and hence the first one is correct here, right. So, just few changes NTA will do, and repeatedly it has asked 3 times this question. Okay. Thank you, Safura. Next question. Yeah, so this is all NTA has asked till now from 2013 to 2021 related to the topic the DNA, including packaging and structure of the polynucleotide chain. Now, let us pick another topic, that is the search for genetic material, and let us see what are the types of questions till now NTA has asked from 2013 to 2021. Get ready for the question. The experimental proof, yes, very simple, very simple. Either NTA will ask about the scientist or will ask about the organism on which these scientists have worked in order to discover or identify who exactly is the genetic material in at least majority of the living organism, and right now we know it is DNA. So, the experimental proof for semi-conservative replication of DNA was first shown in, very simple, anybody would like to give it a try, anyone? Give it a try at least. Anyone? Okay, let me tell you then. Yeah, it is bacterium, very right. Excellent, Safura, the answer is bacterium. Let me tell you which bacterium, it was E. coli. It was E. coli. So, remember students, who did this experiment? Anyone who did this experiment? Michelson and Stahl. Remember this diagram, right? Remember this diagram, this experiment. At least if you would have forgotten the name of the scientist, at least you must remember this image, right? So, this image was related to the experimental proof that was given by Michelson and Stahl on E. coli, where they were trying to prove that DNA replicates semi-conservatively in living organism with the help of E. coli, right. Then further on it was shown on plants and human cells also, but for the very first time in a living organism DNA replicates semi-conservatively was proved by Michelson and Stahl in this experiment, right. And you remember, let me give you a brief, or let me give you a tip exactly how these people concluded that DNA replicates semi-conservatively. So, see your students, this is E. coli takes 20 minutes, like the cycle takes 20 minutes. So, in 20 minutes this is the first generation. After 40 minutes there will be second generation, right. So, remember students, in second generation when centrifugation was done, and the DNA, these scientists were trying to find out the density of the DNA that is possessed by the E. coli that are produced in second generation, then during the experiment in the centrifugation tube they found two density bands. That simply means there is presence of what? There is a presence of a DNA in daughter cells where one strand is parental and another one is newly synthesized, right students. So, they obtain light band and hybrid band. So, if DNA would not have replicated semi-conservatively, then there should be just two strands, one light and one heavy, but instead of getting heavy they got light and hybrid. So, presence of this hybrid density band in the centrifugation tube simply indicates that DNA replicates semi-conservatively, and that is why in some of the daughter cells of E. coli, the daughter DNA has one heavy and one light strand, where this heavy strand represents to the parental generation. I hope this is clear to all of you, and we also did in brief the discussion about this whole experiment where I discussed with you the most important conclusion, right. So, it was a brief revision of this whole topic. Next, yeah, very simple question. It was asked in Neet 2018, right. Although it is a mix, right, although it is a mixed question, it is between two chapters, the informations are given between two chapters. Now, let me help you in getting that. The correct match, Ribozyme is a nucleic acid. Yes students, it is a RNA that acts as an enzyme, right. It is a nucleic acid only that acts as an enzyme. Next, F2 crossed with recessive parent is dihybrid cross? No. In dihybrid cross, we try to understand the inheritance of two genes, right. It is not simply F2 cross with recessive, it is not like that. Next, T.H. Morgan transduction. No, T.H. Morgan actually gave the experimental verification of chromosomal theory of inheritance and also linked with linkage, right. Next is G. Mendel, that means Rager Mendel. No, Mendel did not work on transformation. Griffith worked on transformation. Mendel actually considered as father of genetics, right. So, simple, this one is correct, all others are wrong. Next question, the final proof for DNA as the genetic material came from. See students, Griffith did his transformation experiments, then MacLeod, Avery, McCarty also did their biochemical characterization of the chemical that was present or that was transforming the live R into S type of character in the Griffith experiment. These three scientists worked on that, but still the scientists of their time were not convinced and were not agreeing that DNA is the genetic material for at least majority of the living organism. Then finally who came? Hershey and Chase. They gave an unequivocal proof that DNA is the genetic material with the help of an experiment in which they utilize bacteriophages. Bacteriophages are the viruses that affect or infect bacteria. So, they utilize bacteriophages and E. coli to give the unequivocal proof that DNA is the genetic material. So, who did that? Hershey and Chase. And remember this diagram? Remember this experiment? It was done by Hershey and Chase. And what did they use? They used these bacteriophages. One, these bacteriophages, these are actually two different. Parallelly, you need to analyze the results. In this one, you can see the bacteriophages were grown on radioactive sulfur, and here they were grown on radioactive phosphorus. And you already know that DNA contains phosphorus, hence the DNA of this bacteriophage will become radioactive, and here the protein coat will become radioactive because sulfur is present in the amino acid. And after the result, it was concluded that it is DNA that is passing from virus to E. coli and not the protein coat. Hence, the confusion between DNA and protein was solved, and it was considered that DNA is the one which is considered as, DNA is the one that is getting transferred from bacteriophage to E. coli, hence DNA is the genetic material. Right, Safura, everybody? Chalo, let us see the next question then. Taylor conducted the experiments to prove what? See students, Taylor conducted the experiments to prove semi-conservative mode of chromosome replication. So, see students, very simple, right? If I consider here, yeah. So, what I was explaining in the previous to previous question, yes, this was based on what? This was based on semi-conservative mode of DNA, right? So, similarly, even DNA that is present in chromosome also replicates semi-conservatively, and it was proven by Taylor and his colleagues in Vicia Faber. Simple. First it was done with in the case of E. coli, right? Besselson and Stahl did that, and later on Taylor did that on chromosomes also, and finally concluded that yes, even chromosome replicates semi-conservatively, and the experimental plant or experiment were done on this plant, right? I hope this is clear to all of you. Yes, very right, Safura. Next question, this was asked in Neet 2016 second phase. Next question, a molecule that can act as a genetic material must fulfill the traits given below except. So, you need to find out of these four options which one is not the trait, or which one trait should not be possessed by a molecule in order to act as a genetic material. Can you try this question? Crystal clear. Yes, Safura, thank you for letting me know that. Let me know which one is the right option in this, Safura. What do you think, which one should be the right one according to this question? the factor or the trait or the character that should not be possessed by a molecule in order to act as a genetic material? Yes, it should be C. See students, what is C option? C option is it should be unstable structurally and chemically. No, 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 no. The molecule that need to act as a genetic material should be structurally and chemically stable, and that was in fact the only point on which DNA won the race and RNA was left behind, right. Other three, if I talk about able to express itself in form of Mendelian characters, that means from genetic information there should be formation of some or the other kind of protein that can form enzyme, etc. So, that is done by DNA as well as RNA. DNA forms RNA, RNA form proteins that can act as an enzyme, right. Similarly, RNA without RNA directly, RNA can do this work directly. From RNA directly there is a, with the help of translation process, there is a formation of a enzyme, right, a protein. So, first is applicable to both. Second, it should be able to generate this replica, both DNA and RNA can do that. It should be able to provide a scope for slow changes that are required for evolution. Even this is done by both. Only and only it is the third point where DNA won and RNA lost. So, RNA is actually structurally and chemically unstable in comparison to DNA. So, DNA is more stable structurally as well as chemically when it comes to or in comparison to RNA, and hence DNA is considered as the genetic material. Next question students, transformation was discovered by, transformation principle was given by Griffith. Remember the experiment of Griffith? Do you remember the experiment of Griffith? If you do not remember, then let us do a revision. Very simple. So, see students, what Griffith did. He did this experiment with the help of bacteria, right, pneumococcus bacteria. Remember, these bacteria are responsible for developing pneumonia in the rat, right. So, there are two strains, R strain and S strain. S strain is the virulent strain. S strain can develop a mucus coat on it, and because of this presence of mucus coat, they are virulent, they can cause pneumonia, whereas the R strain is unable to cause the pneumonia, right, inside a living organism. For example, here the rat are used. So, if R strain is injected into the rat, rat will not develop pneumonia because there is absence of that mucus coat over the colony of the R bacteria, right. So, this is what was known before starting the experiment. So, when Griffith conducted the experiment, and in the very first one he injected the live R bacteria, into the rat, rat survived. When he injected the live S bacteria into the mouse died. The mouse died, right. Here it was living, here died, and in this way what here heat killed, right, heat killed, when in this case heat, I should write it again rather, it is looking clumsy over here, even over here, right. What are these? This is heat killed S. So, when heat killed S, so Griffith was somehow able to heat kill the S strain, and when this heat kill S strain was injected into the mouse, mice survived, mouse or mice did not die, right, it was living, right. But in the fourth case where there was like a bulb on situation for Griffith, here what he did, he gave a combination of heat killed S plus live R. So, on giving heat killed S and live R, mouse actually died. How come? In this case heat killed S were not able to develop pneumonia in this rat, but in this case heat killed S was able to develop pneumonia. So, that is the observation, that was the observation from the Griffiths experiment, and what he concluded that, right, what did he concluded, that there was transfer of some transforming principle that got transferred or that got transferred from heat killed S to live R. Because of this transfer, the R live R was able to develop characters like that of S, because of which there was production of a mucus coat over the live R, and hence live R was able to develop pneumonia in this rat, and this rat actually died. So, these are the various observations, right. Whatever I have explained, same is written over here. So, I hope this much is clear to all of you, and with this question we actually revise the entire paragraph of NCERT related to the Griffiths experimental or Griffiths experiment or transformation principle, right. I hope this is clear to all of you. So, now let us take replication question, students. That is just one question that NTA has asked, right, from 2013 to 2020, I can say, even 2021 also I can consider, right. So, just one question that was asked in Neet 2017. During DNA replication, Okazaki fragments are used to elongate. Very simple. Can anybody try this? This is the last question of this session. Safura, I want you to try this. Shubika, if you are there, Sunil, if you are there, please try. Give it a try. It is a good question, in fact, right. So, the answer is very simple, the lagging strand. So, during DNA replication, Okazaki fragments are used to elongate the lagging strand away from the replication fork. So, let us try to see with the help of the image, with the help of the diagram, how this, yes, very right Safura, excellent. Now, with the help of this diagram, we can understand. See students, you remember DNA replication, the main enzyme is DNA dependent DNA polymerase. It has one condition, that it will polymerize only in the direction 5:3. Hence, the strand of the DNA, not to forget, during replication both the strands are going to act as a template. So, the strand of the DNA that has polarity 3:5 on this DNA polymerase will form continuous strand, right. But the strand of the DNA that has polarity 5:3 on this DNA polymerase will discontinuously form the strands of the DNA, because it can polymerize only in 5:3 direction. So, these fragments is known as Okazaki fragments, and these Okazaki fragments will be connected with the help of DNA ligase, and hence there will be formation of a strand known as lagging strand. So, this is what students, this continuously formed strand of a daughter DNA is termed as leading strand, right, and this one is termed as lagging strand. This one is termed as, in fact, lagging strand. Why it is known as lagging strand? Because it is formed discontinuously. These separate fragments, it will be formed in form of fragments, and each fragment will be known as Okazaki fragment, and they will be finally joined, ligated with the help of DNA ligase to form a single strand, a continuous one later on after joining, right. But the synthesis is not continuous. Please remember this point, and also remember, please student, remember that second part of that answer. So, the Okazaki fragments, right, these discontinuous fragments are actually synthesized away from the replication fork. So, this is the direction in which replication fork will move during replication, and the Okazaki fragments are formed away from the replication fork like this, right, and that was the question. If you remember, that was the question. The lagging strand away from the replication fork. I hope this question and explanation both are clear to all of you. That's all from today's session, students. I really hope each one of you enjoyed this session where we discuss all important questions that NTA has asked from Neet 2021 to 2013 related to the DNA topic and search for the genetic material. This was part 1. Do not forget to join the part 2 where I will discuss the remaining previous year question of this chapter. So, tomorrow's session is going to be also very interesting. Do not forget to join that one. Thank you for joining this session, students. Thank you for staying connected, and thank you for participating. Thank you all. Thank you Safura, Sunil. Thank you Shubika. Thank you everyone. Give me a thumbs up if you like me in this session, and do not forget to subscribe Infinity Learn by Sri Chaitanya, and also do not forget to hit the bell icon so that whenever in the next time I am going to come live, you will be notified every time and won't miss any session in future. Bye bye. That's all.